If you don't see, then I would like to invite next speaker, Yuan Wei from Gansu Agriculture University in China. Welcome.
Yeah, I'm here. Yeah.
You can see it?
Yeah.
Now you can see us.
Professors, can you see my PPT?
Yes, can you make full screen?
Oh, okay. Just wait a minute. Is that okay?
Yes.
Yes, it's fine.
Yeah, thank you. Good morning, everyone. I'm Juan, a researcher from Gansu Agricultural University in China. Today, I will introduce some of our recent researches, and it's about the phenolic compositions and antioxidant activities of berries and leaves from Hypocea tibeta and Rhyamicid sinensis produced in Qinghai Tibet Plateau. And I will explain as the following aspects. First, let's see the background of our research. And China is the largest distribution country of Cibaxin in the world, and the total area is about 2.0 million hectares, accounting about for more than 90% of the total area of the world. And there are 7 species and 11 subspecies of Cibaxin have been recognized worldwide until now. And Qinghai Tibet Plateau is the main origin and distribution area of Hypocea rhamboides sinensis and Hypocea tibeta species. And the Hypocea rhamnoides sinensis is a wild Cibaxin subspecies of China with the broadest distribution and maximum production. But Qinghai Tibetan Plateau is the orange and the main distribution areas of this genus. And I will refer to this subspecies is for short RHA in the following explanation. And the plant of RHA is about 2 to 18 miles high, and its leaves are opposite or nearly opposite, and its berry is orange red or dark orange red. And the species of Tibetan is a unique Cibaxin species restricted to Qinghai Tibet Plateau and its adjacent areas. And the plant of TRB, I will refer to Tibeta for TRB in the following explanation. And the plant of TRB is short. It's about 2 to 8 centimeters high. Its leaves are whorled, and its berry is orange red or dark orange red. Especially, it's very obviously at the top of the berry is decorated with brown-black star orn. And I think maybe it's only exists in this species. And now let's see some of our results. And as a commonly used edible plants, all parts of Cibaxin are proved to be rich in source of bioactive compounds like phenolics and carotenoids, organic acid, and so on. So the rich source of biotic compounds endows Cibaxin with various beneficial effects on health, such as antioxidant, antibacterial, anti-cancer, and anti-inflammatory effects. Until now, there is no report about the comparison of phenolic composition and the antioxidant activities of RHA and TRB leaf and berry, which are distributed in Qinghai, Tibet. In our research, the comprehensive phenolic profiles in these two species of Ciboxan leaf, berry, and their quantitative data were investigated. Moreover, its antioxidant activities of phenolic extracts from these two species of Ciboxan berries and leaves were examined with the extracellular assays or the cellular assays. So now let's see some of our results. First, these are the results of total phenolic and flavonoids contents in RHA and TRB leaf and berry. In this figure, we can see that the RHA leaf, this is the leaf results. We can see that RHA leaf possessed more total phenolic and flavonoid contents than TRB leaf. And in berry, we see the similar results. And RHA berry have more total phenolic and flavonoid contents than TRB berry. And more importantly, when we compare these two figures, we can find that the total phenolic and flavonoid contents of leaves were much higher than that of berries. It's about nearly 10 times higher. We can see from this figure, it's about nearly 10 times higher in leaves than in berries. So after estimating the total phenolic and flavonoid contents, the phenolic composition and contents were identified, and there are 11 phenolic subclasses were identified. And we can see this is for the results of leaves, and we can see that benzoic acid derivatives and its in the leaves of RHA is the highest. Is accounting for 68 and 26% of total phenolic compounds in RHA and TRB. And alic acid is the highest polyphenol in the leaves. Meanwhile, flavonols were the highest subclass in the leaves of TRB species, is accounting for 25 and 44% of the total phenolic content in RHA and TRB, among which rutin content is the highest. And 66 polyphenols were identified in the leaves of RHA and TRB. When we compared these two species, there were significant difference in the expression of 42 substances, and the higher content of alic acid. Yes, we can see from here alic acid, gallic acid, mesogallate, and 2,6-dihydroxybiosic acid, and nicotine fluorine, and rutin. Rutin is higher in RHA leaf than in TRB leaf, but procidin B3 is higher in TRB leaf than in RHA leaf. This is the results for the berry, and we can see from here the flavonols were the highest subclass in berries, which accounting for 94% and 64% of total phenols in RHA and TRB berries, among which rutin and narcissin were the highest substances. And the content of catechins in TRB berries is high, accounting for about 31% of the total phenolic contents. Among them, epiglycine is the highest content. And we compared the phenolic compositions and components between these two species berries, and there are 46 polyphenols were identified in the berries of RHA and TRB leaf, and there were significant difference in the expressions of 29 substances, and the higher content of rutin, nasim, and nicotine fluorine, and quinine 3 galactoside, and isoamanitin 3 or glucoside in RHA is higher than in that TRB, but we can see that the epiglycine in TRB berry is higher than that in RHA berry. Then we test the antioxidant activity of phenolic extracts from RHA leaf and berry. First, we can see from this figure, and the ABTS and DPPH scavenging ability and the total reducting ability of RHA was stronger than that of TRB, both in leaves and berries. And the antioxidant capacity antioxidant capacity of leaf is higher than berry. So the above antioxidant activity assays were tested only use chemical assays. However, the above chemical assays could not evaluate the antioxidant performance of tested samples in physiological conditions. So a CAA method was developed in cells. This method could reflect the cellular uptake, distribution, and metabolism of antioxidants under conditions. It could mimic a biological system. So the antioxidant capacity of RHA leaf and berry and TRB leaf and berry was tested with the CAA method. We can see from these figures the antioxidant capacity of RHA leaf and berry were better than TRB leaf and berry at cell level, both in without PBS wash group or PBS wash group. Based on the above results, we can come to the conclusion that the total phenolic and flavonoid contents of RHA and TRB were both present in a high level, and the content in leaf is nearly about 10 times higher than that in berry. Moreover, the total phenolic and flavonoid contents in RHA leaf or berry were higher than that in TRB, and the polyphenol extracts from the leaves or berries of RHA and TRB exerted extraordinary antioxidant activities, both in extracellular and in cellular levels. And the antioxidant effect of RHA is significantly stronger than that of TRB, most probably due to its high total phenolic and flavonoid contents. So we can see that Sebaxin leaf and berry, especially RHA, the RHA subspecies hold a great potential to serve as the resource of natural antioxidant dietary supplement used in food industry. But further researches shall be carried out to investigate the active compounds and that account for the antioxidative activities. Furthermore, the synergistic and individual effects of phenolic compounds should also be revealed in the future research. And based on the above results, we published these two papers. If you are interested, you can email me. And This results most carried on by our two students, and the experiment was supervised by Professor B. And thanks for your attention. Any questions?
Thank you for your presentations. It was very interesting. Yes, we have time for questions. Okay, Thomas was first.
I would like to ask.
Yes, I see. Oh, Virendra, you have a question?
Yes, a very interesting presentation. And, you know, Tibetana grows both in China and India, but in India it grows above 3,800 meter up to 4,500 meter. That is the only woody plant growing at that altitude. Our studies also found that it is less richer in antioxidant compounds, but sometimes I think there must be some compounds which help the plant in surviving at that altitude, and those compounds may be of some use for human health, but that investigation has not been done, and I think it has the future. Presently, this plant is not being used in food, and I think also in China, whether Tibetana has some use in China in food or medicine.
You mean the species of
Tibetana.
Tibetan? Yeah. Sometimes in China it's now used for fruit. Yeah. Not for because its production is not too much. The local people used it as a fruit. Yeah. Maybe I didn't answer the question. Do you ask if the Tibetan species used for?
Yes, I have suggestion that this plant needs lot of investigation, particularly identifying some compounds which may help the plant in surviving at that extreme climatic conditions at high altitude. And those compounds may be of some use. Yeah. That is a suggestion only. Okay, thank you.
Yeah.
Okay, thank you. Then next question could be from Hamid, please. Hamid.
Yeah.
Hamid, I did not hear you.
Daria, maybe we ask
How are you
Nicholas.
doing?
Hamid, can you switch on? Okay, till we're waiting from Hamid, then Nikos, welcome, your question.
Okay, thank you. My question concerning the leaves. What is the processing you are doing to the leaves for the market? I mean, the products that you are the company is making from leaves, except tea. I know that there is products with leaves for tea, but is there any other products that you are producing for the market, for the consumer?
It's a very good question. Yeah, most of the leaves were used for tea, and now we think about to make it as a power. You know, the leaf power used to add some food, such as cake or some other kind of food. So maybe it could make the give the food more flavors. Yeah, make the leaves into a powder, then use the powder to add into food. Yeah.
Okay, thank you.
Thank you.
Now, Hamid.